On the 29th of November 2018, the world woke up to a headline which sounded like science fiction. The first genetically modified humans had been born. The scientist who created them is now in prison, but the technology which made them possible has not gone away. When you hear the term genetically modified, your mind might jump to ideas of designer babies, edited to be taller, prettier, cleverer. However, this was not the case here. Jiankui was the first scientist to use gene editing to disable the CCR5 gene in the embryos of twin girls, Lulu and Nana. The father of the girls was positive for HIV, so this was done in an attempt to confer resistance to the virus. Jiankui used a technique known as CRISPR. During this, a molecule, the guide RNA molecule, binds to the target DNA sequence. This then directs the Cas9 enzyme to bind to and create a double-stranded break in the DNA sequence. This is repaired by the cell repair mechanism, and nucleotides can be replaced or added, either disabling the gene or introducing new DNA sequences. You could think of it a bit like cutting up a piece of paper. Even when you glue it back together, the role of the cell repair mechanism, it will never be exactly the same as it was before. However, since then, Jiankui's experiment has become a global scandal, branded as unsafe, reckless, ethically indefensible. As well as the numerous risks associated with CRISPR technology, this was in part due to the fact that the twin girls were never actually in extreme danger themselves, because HIV cannot be directly inherited through the sperm, and as such was incredibly unlikely to be passed on to the girls until after birth, which could be avoided with small precautionary steps. As well as this, the CCR5 gene is what is known as pleiotropic, meaning it has several important functions within our body. One of these is in reducing the risk of contracting influenza. So, in trying to protect the girls from one virus, he may have accidentally made them more susceptible to another. The issues were not just with what Jiankui did, but also how he did it. He failed to garner sufficient consent for his experiment. The document he submitted was 23 pages long and written in English, not the first language of many of the participants. It also contained many technical terms and abdicated almost all legal responsibility from possible negative outcomes. That's a bit like expecting someone to read all of the terms and conditions in small print, except they're in another language. And instead of being about your new iPhone purchase, it's about your child's DNA. So you might now be thinking to yourself, none of this sounds very hopeful for the future of gene editing. Should we then completely ban the technology? In one survey, 76% of the British population thought not. There already exist many forms of gene therapy which have been carried out in a much more ethically acceptable manner. One of these is mitochondrial replacement therapy, or MRT. During this, the nuclear DNA from the mother's egg cell containing mutated mitochondrial DNA is implanted into a donor's egg cell containing healthy mitochondrial DNA, thus stopping the mutated mitochondrial DNA from being passed on to the child. However, even when carried out in a much more regulated manner, such as this, gene editing forces us to ask ourselves difficult questions. Is there the risk that one day Gene editing will no longer just be used for treating diseases, but also for enhancement, risking creating a two-tiered society with the edited and the unedited. If we are racing to prevent genetically inherited diseases, are we somehow implying that the lives of those already living with these conditions are somehow less valuable? Gene editing is an undeniable part of our future. You may yourself have recently seen about advancements in gene therapy for treating Huntington's disease, a devastating neurological condition. And whilst these don't at the moment involve editing the genes in the embryo before birth, this could one day be a potential solution for eradicating Huntington's altogether. I myself first became interested in this topic through reading the news about gene editing, inspiring me to write my own essay on the subject. The real question is how, as a society, can we use this exciting new technology responsibly? And what risks are we willing to take to make a future without genetically inherited diseases a possibility? Gene editing is no longer just a question for scientists in labs. It's a conversation for all of us. So I ask you to talk about it, learn about it, form your own opinions. When considering a technology that could one day affect all members of our society, it has never been more important that everyone makes an effort to stay informed. Thank you.